Hi all. Have you ever seen bread with green or black fuzzy growth on it? Or wondered how soft bread becomes fluffy while baking? Interestingly, both are caused by fungi. The fuzzy growth is mold, while the organism that helps bread rise is yeast. Even though they belong to the same kingdom, they both look different and they behave differently. So, in this video, let's understand the morphology of molds and yeasts in a simple way. First, remember one important point which I have told you. Molds and yeasts are not different kingdom, both belong to the kingdom fungi. The main difference is in their structure or morphology. Molds they grow as long thread-like filaments called as hyphae, and many hyphae together forms a network called as a mycelium, which gives the molds their fuzzy appearance. Yeasts, on the other hand, are single-celled fungi. Each cell lives independently and usually reproduces by budding. So remember, the easiest way to remember is molds they are filamentous fungi and yeasts they are unicellular fungi. So keep this difference in the mind because it's one of the most commonly asked questions and confusing point. Okay, now let's move on to the next slide. Classification of fungi. Now let's look at the basic characteristics of fungi. Fungi, they are eukaryotic organisms. So they have true nucleus and membrane bound organelles. They cannot make their own food like plants, so instead they absorb nutrients from the surroundings after releasing digestive enzymes. Their cell wall is mainly made up of chitin and glucans, which provide strength and protection. Okay, now based on their morphology, fungi are classified into three groups: molds, yeasts, and dimorphic fungi. So the molds, they are multicellular fungi made of hyphae. Yeasts, they are unicellular fungi that are usually reproduced by budding. And dimorphic fungi, these are the fungi that can exist as both molds and yeast depending on their environmental conditions. So we will study dimorphic fungi in detail later. So now that we know the basic classification, let's move on to the detailed structure of molds. Okay, so see, when molds grow on bread, fruits, or culture media, they appear as fuzzy, cottony, velvety, or powdery colonies, right? So although it looks like one patch, it is actually made up of thousands of microscopic threads, and these threads are called as hyphae. And many hyphae join together to form a network called mycelium. Think of this, it like one thread, it is hyphae, and many threads together, it is mycelium. So this is one of the easiest and most important exam questions. Okay, see the types of mycelium here. The mycelium, they have two main parts. One is vegetative mycelium, which grows inside the substrate and absorb nutrients for the fungus. Okay, aerial mycelium, they grow above the surface and produces spores for reproduction. Now, a simple trick to remember is vegetative, it is for nutrition, and aerial is for reproduction. Now let's see the septate and aseptate hyphae. See, not all hyphae have the same structure. Some have cross walls called as septa, as you can see, while others do not have. So let's talk about septate hyphae. Septate hyphae, they contain cross walls called septa. These septa have tiny pores that allows the cytoplasm and nutrients to move between the cells. So the examples are aspergillus and penicillium. Now let's talk about aseptate or coenocytic hyphae. So these aseptate hyphae, they do not have cross walls, and the cytoplasm is continuous and contain many nuclei. So they are called as coenocytic hyphae. Now the examples are rhizopore and mucor, and remember this simple rule: septate means partitions are present and aseptate means no partitions is present. So this is one of the first features microbiologists observe while studying the fungi under the microscope. Okay, now instead of memorizing names, let's identify them by their appearance. So you see here the aspergillus, they have a round vesicle with chains of conidia, giving it like a dandelion like appearance, right? And talking about the penicillium, they have branch structures that look like a paintbrush. Okay, and rhizopus, it is common bread mold. So it produces spores inside a round sporangium and has a aseptate hyphae. Okay, and mucor looks similar to rhizopus but does not have rhizoids, which helps distinguish the two. Okay, now quick revision. Aspergillus, they look like dandelions. Penicillium, they look like paintbrush. Rhizopus, they are bread mold. And mucor, they have no rhizoids but look similar to the rhizopus. Now we have understood the filamentous fungi in detail. So next let's move on to the second major group, which is yeast, and which are completely different because they exist as single cells. Okay. So unlike molds, yeasts are unicellular fungus, and a single yeast cell is complete organism. They are larger than bacteria but are still microscopic, so they can only be seen under a microscope. So instead of forming true mycelium, the yeast usually appear as round or oval shapes. So as you can see here, now let's look at the important parts of a yeast cell. Now, the cell wall. The outer layer is called as the cell wall, which gives the cell its shape and protection, and it is made up of chitin, glucans, and mannans. Now, the plasma membrane. Below the cell wall is the plasma membrane, which controls the movement of substance in and out of the cell. And a very important point is that the fungal cell membrane contains ergosterol, while human cell membrane they contains cholesterol. And many antifungal drugs, they work by targeting this ergosterol. So easy way to remember is human cells, they have cholesterol, and fungal cells, they have ergosterol. Now talking about the nucleus and organelles, since fungus, they are eukaryotes and they have true nucleus along with mitochondria, vacuoles, and other membrane bound organelles. So in one line, yeasts are unicellular eukaryotic fungus and they usually reproduce by budding. Okay. So students often confuse in these two, like true hyphae, they are long, continuous, tube-like filaments with parallel walls and true septa. Now the pseudo hyphae, they are chains of budding yeast that they remain attached, and they show constrictions where the cells join together. So the easiest trick is true hyphae is straight and pseudo hyphae is pinched. And one important organism that forms pseudo hyphae is Candida albicans. Okay, remember that. Now let's remember the important yeast by their special features. First one, Saccharomyces cerevisiae, also known as baker's yeast. It is used in bread making, baking, brewing, and during fermentation. It produces carbon dioxide and alcohol. Okay, the carbon dioxide that makes the bread soft and fluffy, right? Now let's talk about Candida albicans, and it's an opportunistic pathogen that normally lives in the human body but can cause infections when immunity is low. And it is well known for forming the pseudohyphae, which I told in the previous slide. Now next example is Cryptococcus neoformans. This yeast has a thick polysaccharide capsule which protects it from immune system and increases its pathogenicity. Now in bread, it is Saccharomyces cerevisiae, and pseudohyphae, it is produced by Candida albicans, and Cryptococcus neoformans, they form capsule. Okay, yeah. Now the most common method of reproduction in yeast is budding. So first, a small bud appears on the parent cell. The nucleus divides by mitosis and one daughter nucleus moves into the bud, and the bud grows large and finally separates to form a new cell. Okay, new yeast cell. Okay, now the small mark left behind is known as a bud scar. So repeated bud scars show that the yeast cell have undergone multiple rounds of budding. And some of the yeast also produced by the binary fission, where one parent cells divide into two equal daughter cells. However, this method is less common than budding. Okay, now that we have learned about yeast structure, reproduction, let's move on to how molds reproduce through spores and complete our study of fungal morphology. Okay, now molds, they mainly reproduce asexually by forming spores. And there are two important types you should know. One is Conidia, Conidia are spores produced outside on specialized hyphae called conidiophores, and once matured, they are easily carried by air. The example are Aspergillus and Penicillium. Now talking about sporangiospores. Sporangiospores, they develop inside a sac called the sporangium, and when the sporangium ruptures, the spores are released. Okay, and the example are Rhizopus and Mucor. See, Conidia, it is outside, and sporangiospore, it is inside the sporangium. Now, the sexual spores are formed after meiosis and are important for fungus classification. Fungal classification. The sexual stage is called as telomorph, while the sexual stage is called as anamorph. Now, the three important sexual spores are zygospore. It is produced by fusion of compatible hyphae, thick-walled and resistant. And we have ascospore and then basidiospore. So the ascospore, it is produced inside an ascus, characteristic of ascomycota. And basidiospore, it is produced on a basidium, characteristics of basidiomycota. So the memory trick is zygomycota, zygospores, ascomycota, ascospore, ascus, and basidiomycota, basidiospores, basidium. Now let's talk about dimorphic fungus. Some fungi can exist in two different forms depending on temperature. So these are called as dimorphic fungus, and around 25 degrees Celsius, like means in environment, molds form. Okay, around 25 degree Celsius mold form, and around 37 degrees Celsius yeast form. Okay, the easiest way to remember this is mold in the cold, yeast in the heat. Okay, mold in the cold, yeast in the heat. Now important examples include Histoplasma capsulatum, Blastomyces dermatitidis, and Paracoccidioides brasiliensis. Okay, so these are some of the examples. And then we have Sporothrix shizki, and the exception is Coccidioides and Imitis form spherules in tissues instead of true yeast cells. Okay, now let's quickly compare them. Yeast, they are unicellular. Molds, they are multicellular. Yeast budding, molds by spore formation, and yeast, they have smooth colonies and there is no true mycelium, and in molds they have fuzzy colonies and they have mycelium present. So if you remember these four differences, you will answer the most of the questions. Right, now let's see the laboratory diagnosis of this. Okay, let's finish the commonly used laboratory diagnosis here. First is KOH mount. So it is used for direct microscopic examination. So KOH clears the keratin and other debris, making fungal elements easier to see. Okay, then we see through the microscope. And then we have lactophenol cotton blue. It is used to stain fungal hyphae and spores for microscopic identification. Then we have SDA, which is Sabaron dextrose agar. It is a standard culture medium for fungus, and its acidic pH helps suppress the bacterial growth. Okay, now quick revision: KOH, direct microscopy; LPCB, which is lactophenol cotton blue; fungal stain, SDA, Sabaron dextrose agar. It is a culture medium. Okay, now let's revise the important points. Molds are multicellular, yeasts are unicellular. Hyphae together they form mycelium. Vegetative mycelium absorb nutrients. Aerial mycelium produces spores. Now septate hyphae have septa, aseptate hyphae do not. Yeast, they mainly reproduce by budding. Conidia are produced outside. Sporangiospores are produced inside the sporangium. Okay, true hyphae are straight. Pseudo hyphae have constrictions. Mold in the cold, yeast in the heat. Okay, now that's the complete overview of morphology of molds and yeast. If you understood these concepts, fungal identification practicals and university exams will be much more easier for you people. If this video helped you, share it with your classmates and subscribe to Decode Microbes for more simple exam oriented microbiology lessons. Thank you for watching and see you in the next video.